Welcome back to physiology lectures. So today we'll be looking at excitable tissue, and you know to say under excitable tissue, we have nerve cell and the muscle cell. So we've already discussed the nerve cell. So now we'll start looking at the muscle cell as an example of excitable tissue. So the muscle cells, they are capable of propagating an action potential. They can get stimulated by the motor neurons, which are nerve cells. So remember the previous lecture, we are looking at transmission across the synapse. So synaptic transmission, of which we said the motor neuron that is innervating the muscle cell, it can fire an action potential that will bring about exocytosis of neurotransmitter. At the neuromuscular junction, we have acetylcholine. That acetylcholine, once it has been exocytosed because of the stimulus in form of an action potential, this action potential that is being propagated towards the axon terminals is going to stimulate the opening of calcium variegated ion channels that will bring about influx of calcium, because the concentration of calcium is more in the extracellular as compared to intracellular fluid compartment. So there will be influx of calcium. That calcium will go and bind to V snare, T snare, synaptobrevin syntaxing interaction. It will facilitate exocytosis of a neurotransmitter, acetylcholine. And that acetylcholine will go and bind to acetylcholine receptors, stimulating the motor end plate to a threshold, and then to fire an action potential that will be propagated down the sarcolemma. The sarcolemma is simply the plasma membrane of a muscle cell. So we looked at all that. So now we need to appreciate muscle physiology. There are a couple of lectures in muscle physiology, but we need to start by looking at the general muscle morphology. So the structure of the muscles. This is more of anatomy, but you need to appreciate anatomy for you to be able to apply the physiology. For the physiology, you're looking at the function of the muscle cell. So before we discuss the function, you need to understand how a muscle cell looks like. That's why we are starting with the general morphology of muscle. Okay, so there are different types of muscles. By now, you understand that we have cardiac muscle, the muscle fibers that are found in the heart itself. So they are called the myocardium. So cardiac muscle cells. Then we have smooth muscle cells, two different types of smooth muscle cells. Single unit smooth muscle cell, which is also referred to as visceral smooth muscle cells. Then we have the multi-unit smooth muscle cells. So there will be differences between single unit and multi-unit smooth muscle fibers. Then we also have skeletal muscle fibers. So in terms of structure, the skeletal muscle fibers and cardiac muscle fibers, they are similar, but there are some minor differences that we'll be looking at today. So types of muscles. So like I said, there are three different types of muscles: the cardiac muscle cell, the skeletal muscle cell, and smooth muscle cell. So the cardiac muscle cell and skeletal muscle cells, they are similar in terms of structure because they are both striated muscles. So when you're looking at these muscles here, they appear to have some striations or some lines, which are called striations. So they are called striated muscles. And these striations is due to the arrangement of the myofilaments that are found within the muscles. So they are arranged in a particular manner where you have thin filament and thick filament. So that is arranged to form a sarcomere. So those sarcomeres will result into giving this appearance, the striated appearance to the muscle, as opposed to the smooth muscle cells. They don't contain sarcomeres, so if there are no sarcomeres, under microscope, the muscle just appear to be smooth, hence the name smooth muscle. So the general properties of muscles, there are four major properties of the muscle. So any muscle, it will have these properties. So the first one is excitability. That's why we call it excitable tissue, because the muscle cell, they can get excited with electricity, which are called nerve impulses or action potentials that will be propagated to the muscle cell. So they can also get excited. They have the capacity to respond to a stimulus. You know, that stimulus could be an action potential, so they can get excited. So that is called excitability. The capacity of the muscle to respond to a stimulus, and these stimulus are action potentials. So they can get excited with action potentials. Then the second property is contractility. They are muscles, right? So they need to contract, to say, as muscles are contracting, they will produce a mechanical work outside. So I'm able to move from point A to point B. You only see me moving, but what is doing the work there are the muscles. As the muscles are contracting, they will be able to change some angles of the joint to enable me to move from point A to point B. So they have this property which is called contractility. And the contractility is the ability of the muscle to shorten and generate a pulling force. So they are able to shorten and generate a pulling force that will produce mechanical work outside the body that you can see that somebody is working, somebody is walking, somebody is doing something. So that is due to contractility. But it doesn't necessarily mean that when the muscle is contracting, it's going to shorten. Sometimes you have a situation whereby the muscle is contracting, but it's lengthening. So look at all that. Then the other property is extensibility. Extensibility, it means that the muscle can be stretched. So it can be extended. So when you stretch a muscle, it's going to extend. So that property is called extensibility. So the ability of the muscle to stretch. So once you pull on the muscle, it can be stretched. Why? It's because you have elastic component to the muscle. Those connective tissue, the fascia that are covering the muscle, the epimysium, the endomysium, the perimysium, once you pull on them, they can be stretched. Then after extensibility, we have elasticity. Elasticity is the opposite to extensibility. So you find that the muscle has got the ability to recall to its original resting length. So when you stretch a muscle and then you release it, it's going to recall, or it's going to go back into the resting length of that particular muscle. So that is called elasticity. So it can be elasticated like a rubber band. So you're able to stretch it. When you release it, it will go back to the original length. So that is called elasticity. The ability of the muscle to recall to original resting length after it has been stretched. So at least remember these 4 properties of a muscle. Moving on, just adding a bit of information to each type of the muscle. So the general description of muscles. So the first, let's start by looking at classifications of muscles. So I've already told you to say that there are 3 major types of muscles. So how can you classify muscles? You can classify muscles based on the appearance of the muscle. So let's start with the gross appearance of the muscle. When you're just looking at the muscle grossly, are you able to tell? Yes. Even when you go to the butchery to buy meat, you are able to tell them, say, can you give me steak? What you're buying there are skeletal muscles. Sometimes you can tell them to say, can you give me intestines? Once you're given intestines, you'll be able to tell that these are intestines because you're able to tell that those are smooth muscles. Then sometimes you can tell them to say, I want a heart. So they will give you heart muscles. So you're able to tell this is a heart. So based on gross structure, you are able to tell the 3 different types of muscles: skeletal muscle, smooth muscle, and cardiac muscle. Then according to the microscopic structure, you can also differentiate muscles into striated and unstriated. So according to the microscopic structure, so you have striated muscles, muscles that will appear to have some striations or striped muscles. So an example there, skeletal muscle and cardiac muscle. Skeletal muscle and cardiac muscle are striated muscles because when you look at them under microscope, you'll be able to appreciate the structure of the sarcomere. So you have the thin filament and the thick filament, the dark region of the muscle and the light region of the muscle, and it'll give you those striations. So only 2 muscles are classified as striated muscles. So skeletal muscle and cardiac muscle. Then if they don't have those striations, they are referred to as unstriated muscle. So a good example of unstriated muscle are smooth muscles. So that's why they're called smooth muscles, because they don't have those striations. So they are called unstriated muscles. Then you can also classify muscles based on function. So the function of the muscle, is it being used in a voluntary action or involuntary action? So if you need to volunteer for you to cause that muscle to contract, they are referred to as voluntary muscles, like skeletal muscles. Whatever I do with my skeletal muscles, I need to send impulse from my conscious, so from my brain, the higher center in the brain. I will send information in form of action potentials. So those motor neurons that are innervating the skeletal muscles, they need to fire an action potential to stimulate the voluntary muscles to contract. So I'm using my conscious. So I need to volunteer to make that kind of movement. So they are classified as voluntary muscles. So skeletal muscles are voluntary muscles. Then we also have involuntary muscles. So involuntary muscles, I don't have control over them. So it simply means that they are being controlled by autonomic nervous system or maybe hormonal regulation of the muscles. So they can respond to certain hormones, they can respond to autonomic nervous system, the parasympathetic and the sympathetic, that is innervating those smooth muscles. And on top of that, certain organs, they have their own intrinsic nervous supply, like the GIT, it has got its own nervous supply. So you have the submucosal plexus, the myenteric plexus that can also stimulate the muscles of the GIT. Those are basically smooth muscle cells. So these are different types of muscles and how you can classify them based on gross structure, microscopic structure, or the function of the muscle. So adding more information to each type of the muscle, starting with the skeletal muscle. So from the name itself, it means that the muscle fibers, they are attaching to the bones. So the muscle fibers are attaching to the bones. They make up 40% of the body weight. Then they are responsible for locomotion, facial expressions, posture, respiratory movements, and other types of body movements. So because these are voluntary muscles, you find that they're the ones that are involved in locomotion, the movement of the skeleton, and also facial expressions. By looking at you, I'm able to tell whether you are happy or you're unhappy, because there are certain muscles on your face that will contract when you're unhappy. Then they will give you that facial expression of sadness. So somebody is able to tell. Then posture, you're able to stand for a long period of time. Why? It's because you're using skeletal muscles to support you against the gravitational force. So you find that you'll be able to sustain your posture. Respiratory movement, you're talking of respiratory mechanics, the breathing mechanics. You have the intercostal muscles, which are basically skeletal muscles that are involved in contraction, relaxation. So you know that respiratory system is supported by skeletal muscles as well. Then there are also other body movements that are not specified here. Then in terms of function, they are voluntary. So they are voluntary in action. They are controlled by somatic motor neurons. So you have somatic motor neurons from the central nervous system, which is the brain and the spinal cord, that will innervate the skeletal muscles. So these neurons are called lower motor neurons. So you have the upper motor neurons from the brain, the spinal cord, then you have the lower motor neurons from the spinal cord all the way to the peripheral structures, to the muscles. But here I'm telling you to say that only skeletal muscle cells will make up 40% of the body weight. So the question really here is, is the body only made up of skeletal muscles and total body water? Because I told you to say total body water is 40% of your body weight. But here I am again telling you to say skeletal muscles only is 40%. So some students might get confused here to say, does it mean that the human body is just composed of water and muscles? It doesn't mean that. But in this 40%, there's some amount of water that is found in the muscle cells. So you find that most of the water is found in the muscle cell. So the muscles, they're contributing 40% of your body weight, of which this 40% may be 39% or 38% is actually water. So you don't get confused there. Smooth muscle cells are found in the walls of oral organs like blood vessels, the eye, the glands, the uterus, the skin. So you find a lot of smooth muscle cells there. You don't have control over them. So some function of smooth muscle cells, they are there to propel urine. Maybe in the urinary system, you have smooth muscle cells in the ureters, urethra, you have smooth muscle cells. So when those smooth muscle cells are contracting, they will help to propel the urine. So to propel urine, sometimes to mix food in the digestive tract. So in the gastrointestinal tract system, the gut system or the intestines, there is contraction of smooth muscle cells. That will bring about peristaltic contractions and mixing contractions in the GIT. So that is the function of smooth muscle cells. Then they are also involved in dilating or constricting the pupil. So, you know, the eye pupil can dilate or it can be constricted depending on the light that is entering the eye. So if there's too much light, you find that they are going to constrict to minimize the amount of light that is entering the eye. If it's too dark, they're going to dilate to allow you to see things in the dark. Okay, so those are smooth muscle cells. So the iris muscle of the eye, it's basically smooth muscle cells. And then regulating blood flow. Vasoconstriction, vasodilatation of blood vessels is facilitated by smooth muscle cells. So if the smooth muscle cells in the blood vessels are contracting, they'll bring about vasoconstriction, minimizing the amount of blood that is approaching an organ. If you have relaxation of smooth muscle cells, you have more of vasodilatation, so it will increase the blood flow to the organs. So smooth muscle cells, they are found in whole organs. So in some location of smooth muscle cells, they can have autorhythmic or they are autorhythmic. Autorhythmic, it means there is an activity of pacemaker. So some of them, they can function as pacemaker cells. They are capable of generating their own action potentials. That's why the GIT and also the heart, they are autorhythmic. It means that they contain pacemaker cells. And we said the pacemaker cells, they don't have a stable resting membrane potential. It's always changing. So if there's depolarization to the threshold, they'll fire an action potential that can stimulate the myocardium if it's in the heart, or they can stimulate the smooth muscle cells if it's in the GIT or the stomach or small intestines, large intestines. We find those pacemaker cells, which are called interstitial cells of Kagel. So you appreciate later on, once we start discussing GIT physiology or physiology of the GIT. Cardiac muscle cells, you know, they are the major source of movement of blood. So blood is being pumped by the ventricles. So you have the two atria chambers, and then down there we have the two ventricles. So when the ventricles are contracting, they are going to squeeze blood into the aorta and the pulmonary circulation so that blood can be transported to the lungs for oxygenation or gaseous exchange. And then once there is gaseous exchange, the blood will return back to the heart via the pulmonary veins. So you have the pulmonary artery and the pulmonary veins. So once it returns to the heart, it will be pumped into the aorta, then it goes to systemic circulation. So what is facilitating the movement of blood? The myocardium, the cardiac muscle cells in the ventricles of the heart. Then they are controlled involuntarily by endocrine, just like the smooth muscle cells. They can be controlled by the endocrine and autonomic nervous system. So the autonomic nervous system, we have the parasympathetic and the sympathetic branch of autonomic nervous system. The two branches, they work antagonistically, meaning that they will have the opposite function. One can be stimulatory, the other one can be inhibitory. So those are antagonistic because even the neurotransmitters or the neurohormones that are going to be released there, acetylcholine and norepinephrine, they'll have antagonistic effect on most tissues. So in appearance, the skeletal muscles are striated. They are innervated by motor neurons. So you can see a motor neuron that is innervating the skeletal muscle fibers. Then in terms of appearance, they are striated, then they have a multiple nuclei inside. So you have multiple nuclei inside. So they are referred to as multinucleated muscle fibers. So they will have a lot of nuclei inside, which makes sense because A muscle fiber, it's a long cell. Maybe it can start from the tendon, which is the origin, to the insertion of the muscle cell to another bone. So you have origin and insertion. So they are long cells. So because they are long cells, it's very difficult to have just one nuclei to control cellular activities. So God just designed them. They should have contained a lot of nuclei in them to facilitate control of cellular activities. Smooth muscle cells, because you don't have that kind of arrangement, you don't have sarcomeres in smooth muscle cells, so they will appear smooth. But you have two different types of smooth muscle cells. We have the single unit smooth muscle cells. In a single unit smooth muscle cells or visceral smooth muscle cells, they do have a lot of gap junctions, which you can see in this diagram here, that they have a lot of gap junctions. So if a few cells are stimulated, they are able to communicate via gap junctions. So because the cells, they do have gap junctions, it's not necessary for the autonomic nervous system to innervate each nerve fiber. So only few muscle fibers are innervated by velocities. So you can see velocities, these are expansion of the parasympathetic and the sympathetic that are responsible to release the neurotransmitters that can either stimulate or inhibit the muscle cells. So you have different types of neurotransmitters, like we discussed. They are inhibitory neurotransmitters, they're stimulatory neurotransmitters. So these are the ones now that can stimulate or inhibit the smooth muscle cells. But other smooth muscle cells, they don't have a lot of gap junctions, like the multi-unit smooth muscle cells. The multi-unit smooth muscle cells, they don't have a lot of gap junctions. So you find that each and every muscle fiber is innervated by a velocity. So it means for them to contract, they're not communicating via gap junctions because they're not there. So each and every cell has to be stimulated by a velocity. Then we also have the cardiac muscle cells in structure. They are striated. So in appearance, it looks like that of a skeletal muscle. But we realize that the cardiac muscle cells, they can form branches. So you can see the branching off of muscle fibers, which you can't appreciate in the skeletal muscle cells. So the heart is round like that. So those muscle fibers, they'll form branch to facilitate the formation of the ventricles, so that as they are contracting, they are going to squeeze blood out of the ventricles. So it has got branches, and each muscle fiber is separated by intercalated discs. So in between the muscle fibers in the myocardium, we have intercalated discs there. So on the intercalated discs, you have cell junctions. That's where you can also find gap junctions. You can also find other things like proteins that are anchoring one cell to another cell at the intercalated disc there. So just know that the cardiac muscle fiber, they are shorter as compared to skeletal muscle fibers, because in a cardiac muscle fiber, the muscle fibers are separated by the intercalated discs, which you can see here. And most of these muscle fibers, they will only have one nucleus. So they will have just one nucleus, but sometimes you can find one muscle fiber that can contain maybe up to 2 nuclei inside, but most of them, they only have one nuclei. Okay, so those are just some minor differences between the skeletal muscle and cardiac muscle, but under normal circumstances, when you're looking at them under microscope, they will appear to have some similarities as opposed to that of smooth muscle cells. So what are the functions of skeletal muscle cells? So now we just biased towards skeletal muscle cells. So what are the functions of skeletal muscle cells? So function number one, skeletal movement. So it's involved in locomotion. So the movement of the skeleton, because there are muscles that are attaching to the bones via a tendon. So if a muscle is contracting, it's going to pull on a tendon and then to pull on the bone. That will cause change in angle of the joints. So if the angle of the joints are changing, that can produce a movement. So the first function of skeletal muscle cells is skeletal movement. Then the second function, posture and body position. So posture, so for you to maintain posture, you're using skeletal muscles. For you to maintain body position, you're also using skeletal muscles. then support of soft tissue. So the skeletal muscles can also support soft tissues. For instance, you have the intestines, the visceral organs of the intestines that are within the abdominal cavity. But the abdominal cavity is covered by skeletal muscles. So the skeletal muscles outside, they are supporting the smooth muscles or a smooth tissue that is inside in the abdominal organ, abdominal cavity. So those visceral organs in the abdominal cavity, they are protected by skeletal muscles. Then skeletal muscles, they're involved in guarding of entrances and exits. So guarding of entrances. So entrances like the oral cavity, you know, in the lips here, we have skeletal muscles. That's why I'm able to communicate with you. I'm able to speak because I'm continuously sending action potentials that are causing contraction of the muscles of the lips and also contraction of the muscles of the larynx that is producing a particular sound for you to get what I'm saying. So guarding of entrances to the body. So all the entrances to the body, they are guarded by skeletal muscles that can function as sphincter muscles as well. So another example, exits, the anus. The anus is composed of the internal sphincter muscle and the external sphincter muscle. So we have the internal anal sphincter muscles and external anal sphincter muscles. The external anal sphincter muscles are skeletal muscles. So you have control over them. So it's very important that certain exits should have skeletal muscles because you can control the opening and the closing of those exits. Maintenance of body temperature. So when it's very cold, right now it's very hot, so you're not having much of muscle activity. But when it's very cold, you can have muscle activity in form of shivering. You start shivering, so there is muscle contraction and relaxation. That will result into a lot of metabolism that will end up producing heat. And also the friction between the muscle fibers can also produce a bit of heat. So the skeletal muscle fibers or skeletal muscles, they're involved in thermoregulation, so maintenance of body temperature. When it's cold or when it's hot, there are certain mechanisms that will be triggered that will bring about thermoregulation of the body. So the nerve and blood supply to the skeletal muscles. Skeletal muscles are controlled voluntarily. So if they are controlled voluntarily, it means that they need to be innervated by somatic motor neurons. So there is need for innervation there. Then on top of that, you know, to say the muscles will contain a lot of mitochondria. So if they contain a lot of mitochondria because they are an example of high energy utilizing cells, so they use a lot of energy that is coming in form of ATP. So that ATP is being produced within the mitochondria. So the inner membrane of the mitochondria, the electron transport chain that will convert some energy into ATP, and that ATP is required for muscle activities. Be it muscle contraction and relaxation, it will require ATP. So you know to say in the inner membrane, oxygen is required to function as the final electron acceptor. So if the muscles, they have a lot of mitochondria, it means that they will also have blood supply to the muscles to transport the oxygen that is required and also to transport nutrients that could be required by the skeletal muscle fibers. So skeletal muscles are rich in nerves and blood supply for those reasons that I've already mentioned. But the communication between a muscle cell and the nerve cell, it's via the neuromuscular junction. So you have the chemical communication at a synapse. So at the synapse or the neuromuscular junction, that is where you have communication between a motor neuron, which is a nerve, and a muscle fiber. So there will be release of acetylcholine as a neurotransmitter. So we have those axon terminals. The axon terminals, they're going to communicate with the motor end plate of the muscle. But most of these capillaries will be coiled capillaries. So why do you have coiled capillaries? They'll be able to adapt to changes in the length of the muscle fiber. Remember, the muscle fiber can shorten or it can lengthen. So if we have blood capillaries that we are not able to adapt to those changes, you find that they will start breaking. Okay, so God just designed them in such a way that there is room for them to adapt. If the muscle fiber is lengthening, even the capillaries, they can lengthen. So if the muscle fiber is contracting, then the blood capillaries will become coiled to adapt to that. But again, when you start exercising your body, maybe you start visiting or frequenting the gym to lift those metals so that you have more of hypertrophy of the muscles. As the muscles are increasing, even the blood supply is also going to increase. So even the blood supply is going to adapt to that. So you can see this individual is exercising a lot and then the muscle mass has increased. Doesn't mean that the number of muscle cells are increasing, but they are just increasing in the diameter, which is called hypertrophy. Hypertrophy. So if you have hypertrophied muscles, even the blood supply is going to increase to that muscle because the demand for that muscle has increased, which makes sense. Okay, so the blood system can also adapt to the changes that are taking place in the muscles. Some microanatomy of skeletal muscle fibers. So microanatomy of skeletal muscle fibers, when you are discussing muscle physiology, there are certain terminologies that you're supposed to use. So the vocabulary that you're supposed to use as you are discussing muscle physiology. So the skeletal muscle fiber, which is also called the myofiber. So the muscle fiber is called the myofiber. But the plasma membrane of the muscle is referred to as the sarcolemma. So the sarcolemma is the plasma membrane of the muscle fiber. Then the cytoplasm of the muscle fiber is referred to as sarcoplasm. Sarcoplasm. So sarcoplasm is the cytoplasm of the muscle fiber. Then the endoplasmic reticulum in muscles, they are called sarcoplasmic reticulum. So the sarcoplasmic reticulum are the endoplasmic reticulum of the muscles. Then you have the myofilament, like the cytoskeleton that are found in other cells. In the muscles, they are referred to as the myofilaments. So the myofilaments are cytoskeleton of the muscles. So they will be arranged in a particular manner. They will form the myofibrils. So the myofibrils are composed of the myofilaments, which are the cytoskeleton of the muscles. Okay. So these are some of the terminologies that you're supposed to use as you are discussing muscle physiology. This is just a cross-section of a muscle fiber. So you can see there are multiple nuclei within the muscle fiber. They are located on the peripheral of the muscle fiber. Inside the muscle fiber, you have the myofibrils shown in red. Then you have the mitochondrion or the mitochondria. So you have a lot of these mitochondria. So you need a lot of energy for muscle activity. So when you expand on the myofibrils, you can see that you have this kind of arrangement that is composed of the myofilaments, the thin filament and the thick filament. I'll explain what it means. The skeletal muscle structure is composed of muscle cells or myofibers and also connective tissue, blood supply, and innervation. I've already said that. So the fibers are long, cylindrical, and multinucleated. So the fibers are multinucleated. It means that they have a lot of nuclei inside them. So they tend to have a smaller diameter in small muscles and a larger in larger muscles. So dependent on the type of the muscle you're looking at. Is it a bigger muscle? It means that the diameter of those muscle fibers are bigger. If it's a small muscle, then the diameter of those muscle fibers are smaller. So they're going to develop from myoblasts, but the numbers remain constant. So when the muscle mass is increasing, it doesn't mean that the number of the muscle fibers is increasing. No, it simply means that the diameter of those muscle fibers are increasing because their numbers will remain constant. In appearance, they are striated. The nuclei are they are located on the peripheral of the muscle fiber. This diagram is also summarizing the structure of the muscle, starting from the biggest structure going to the smallest unit of the muscle. So the whole muscle is composed of a lot of fasciculars or fasciculi. So you can see the muscle. A muscle is a group of fasciculi. So singular is called fasciculus, or a fascicle. Then plural, they are called fascicles or fasciculi, So a whole muscle is composed of a fasciculus, a group of fasciculi or fascicles, group of fascicles. Then those fascicles, inside them, they are composed of the muscle fiber. So you can see inside the fasciculus, it's composed of different muscle fibers. These are individual muscle cells now. So a group of individual muscle cells, they will form a fasciculus. And the muscle fibers, they are composed of myofibrils. And those myofibrils, they are composed of the myofilaments that are arranged in a particular structure to form a sarcomere. So you can see a sarcomere there. So a sarcomere is just a special arrangement of the myofilaments that are forming the myofibrils. Okay, so this is just structure, starting from the largest going to the smallest unit of the muscle. So the smallest unit of the muscle is a sarcomere. A sarcomere is composed of myofilaments. So the thin filament and the thick filament. So an example of thick filament is myosin. Thin filament is actintropomyosin and troponin complex. Okay, so when a sarcomere is viewed under electron microscope, you're going to see dark and light bands. Dark and light bands. So why are you seeing the dark and light band? Is because of the special arrangement of the myofilaments. So there is a special arrangement of the myofilaments. Okay, so this is still the structure of skeletal muscle cells. So they are covered with connective tissue, which are called fascia. So when you cut under the skin, you won't expose the red muscles immediately. What you're going to see is a connective tissue, which is called fascia, that is covering the muscle. When you cut that whitish connective tissue, that's when you can see the red muscles under the fascia. So the outermost layer of the muscles, they are covered by a connective tissue, which is referred to as epimysium. So epimysium becomes the muscle sheath which fuses with the tendon. So the epimysium is continuous with the tendon that will attach to the bone. Then inside the epimysium, you have the fascicles. The fascicles, they are also covered in another connective tissue. That connective tissue is called the perimysium. So a fascicle is wrapped by the perimysium. So what is covering the fascicle? Perimysium. A fascicle is a group of muscle fibers. So you can see within a fascicle, you have these muscle fibers. And the muscle fibers are covered by another connective tissue, which is called the endomysium. So endomysium are covering individual muscle cells. So they are covering the muscle fiber. So endo is the innermost, peri is after endo, and then the outermost is called the epi. So endo, peri, epi, mysium, those are coverings that are covering the muscle. Okay. Same diagram, so we make progress. So a sarcomere is a basic contractile unit of the muscle. So the basic structural contractile unit of a muscle is a sarcomere. If you want to understand the contraction and relaxation of the muscle, you need to understand the structure of the sarcomere and the function of this sarcomere, because that's the basic contractile unit of a muscle. So a mechanism of skeletal muscle contraction, you need to understand the sarcomere. That's why we are starting with the structure. So remember, we said when you are viewing the sarcomere under microscope, electronic microscope, you're going to see dark portions and light portions, which are referred to as dark and light bands. So why do you have dark and light bands? So in the letter light, there's an I here. So that I is I band, but it doesn't mean that this I is coming from the letter light here in the word light. No, The letter I comes from the way this part of the muscle is going to respond to polarized light. So it's going to respond isotropically. So it's responding isotropically. Iso means the same. So isotropically. So you can see here, isotropic, it has got I. It starts with an I. So this is called the I band. So the I band, this is the region of the thin filament that does not overlap the thick filament. So I'll explain the sarcomere. So where you have the I band, you only appreciate the thin filaments that are not overlapping with the thick filament. Because of that, they will be lighter in appearance. Hence, the I band represents the region of the thin filament that does not overlap with the thick filament. Then the A band, the word A, comes from the way this part of the muscle is going to respond to polarized light. So it's going to respond anisotropically. So you can see anisotropic. So anisotropic starts with A. So that A is the one that will represent the A band. So the A band corresponds to the entire length of the thick filament. So the entire length of the thick filament is called the A band. So these are the darker portions as opposed to the light portions of the I band that are representing the area of the thin filament that is not overlapping with the thick filament. So always remember that. Then within the sarcomere, at the center of the sarcomere, there's a location which is called the H zone. So the H zone is a region of the thick filament that does not overlap with the thin filament. So you only have the thick filament. So it's called the H zone. So it might not make sense here, but once I share the diagram, it will make much sense. So here it is. So this is a sarcomere. A sarcomere is the basic contractile unit of the muscle. It's the distance from one Z line to the next Z line. So within this sarcomere, there are points where they will appear darker. So you can see a darker point here at the center of the sarcomere. Then on the periphery of the sarcomere, you have light areas. So you have the I band or the light band. Then you have the A band or the dark band where it's darker. So this arrangement of the sarcomere is due to the arrangement of the myofilaments, which are thin and thick myofilaments. So the thick myofilaments are shown at the center of the sarcomere in blue here. So this is the entire length of the thick filament shown in blue. So it will correspond to the A band. So you can see the A band is the entire length of the thick filament, which is myosin. Then you have the I band. The I band is a portion whereby you have the thin filament that is not overlapping with the thick filament. So you only have the thin filament not overlapping with the thick filament. This is called the I band. Then at the center of the sarcomere, you have this point, which is called the H zone. The H zone is a location whereby you only have the thick filament that is not overlapping with the thin filament. So it's called the H zone at the center here, H zone. So the thin filament are anchored to one another by a protein, which is called the M line. So you have the M line at the center here. When you check the diagram on top of us here, you can see that there's also a darker point within the H zone. There's a line there, which is called the M line. So at the M line, you have proteins that are anchoring one thick filament to the next one. So it will be a bit darker. So it's called the M line. M line. So in terms of sarcomere contraction, the sarcomere, the Z lines will be pulled towards the M line. So there will be shortening of the entire sarcomere. So the muscle fiber is going to shorten. So this is just the basic unit of a muscle. It's called a sarcomere. Okay, so a bit of information on the myofilaments, just a bit of information on the myofilaments. So we have two types. So two types of myofilaments in the sarcomere are the thin and thick myofilaments. So the thick myofilaments are made up of myosin. So in muscle, there is myosin 2 with two globular heads in the tail. So it's an example of molecular motors. Remember when we are looking at molecular motors, we said we have molecular motor that can move on microtubules, of which we say there are of two types, the kinesin and the dynein. The kinesin moving to the positive end of the microtubule, and the kinesin is moving to the positive end of microtubule, and the dynein, which is moving to the negative end of microtubule. So the microtubule has got two ends, the assembly end, the positive end, and disassembly end, the negative end. So you know that. Then we had another molecular motor that was moving on actin, the actin-based molecular motor, of which we said an example is myosin. That's why it's coming here. So this myosin is two globular heads. It's a double-headed molecule that can move towards the positive end of actin. So you'll see the interaction between actin and myosin later on. But this is an example of the thick filament. It's called myosin. So in muscles, we have myosin 2. Then the thin filament are made up of three different types of proteins. We have actin, the tropomyosin, and the troponin. So the actin, they are globular actin that can polymerize in vivo to form a filamentous actin. And then you also have a filamentous tropomyosin, which is more of a regulatory protein because it will be hiding the binding sites on actin to inhibit the interaction between actin and myosin. Then the troponin is the major regulatory protein because it's the one that will cause the tropomyosin to move away from the binding site so that there is exposure of the binding site to bring about interaction between actin and myosin. So these are referred to as thin filaments. So actin, tropomyosin, and troponin complex. So the myosin filaments, we said it's a double-headed protein. So you can see the two heads that are connected by the flexible hinge region. So you can see the hinge region of the neck that is connected to the tail. Then the tail, they are connected to the M-line. So here they are shown in purple in this diagram. This is a sarcomere. One Z line to the next Z line, and then you have the thin filament shown in green, then the thick filament shown in purple here. So you can see the heads of myosin that is connected to the tail via the neck. Then the tail, they are connected to the M-line. That's why at the center is a bit darker because we have the M-line. So they are connected to the M-line. Then the thin filaments, they are connected to Z lines by a protein which is called the alpha actinin protein. So we can see that the thin filament, they are directly connected to the Z line or to the Z line. Then at the same time, the thick filament, which is myosin, is somehow also connected to the Z line via a protein which is called titin. So I'll show you the protein. But here I just want you to appreciate the structure of the thick filament, which is myosin. So it has got two globular heads that are connected to the tail via the neck. Then the tail, they are connecting to the M-line. So you have a lot of these myosin heads that are protruding from the thick filament. Then they will be able to interact with actin once the binding sites has been exposed, okay? So those were the thick filaments, and then the thin filaments, you have actin, which you can see here. Actin has got a binding site for myosin, but that interaction is inhibited because of the filamentous tropomyosin protein that is covering the binding sites. Then on different segment of the tropomyosin, you have the troponin complex. The troponin complex is the regulatory protein because it contains a binding site for calcium. So calcium can come and bind to troponin C, causing the tropomyosin to move away, exposing the binding sites, and then there will be interaction between actin and myosin. Okay, so the thick filaments, it's myosin. I've already told you the arrangement of myosin or the structure of myosin. So you have two globular heads, and the hinge region that is flexible, it can change. It's connected to the tail, and the tail is connected to the M-line. So you can see that it's anchored to the M-line. So the myosin heads, they are able to interact with actin. At the center here, you have the H zone. Then at the center of the H zone, you have the M-line. Then the thick filament, which is shown in blue here, is connected to the Z line by a protein which is called titin. So you can see the titin protein that is anchoring the myosin to the Z line. So there's that structural support. Okay, so once there's hydrolysis of ATP, it can cause the neck to change. So it can it can reduce or it can pivot. So the power stroke will cause the myosin heads to pivot, and that will result into a movement. We'll discuss that when we start looking at the actual mechanism of contraction. So the thing, myofilaments. So we have three there. We said actin, tropomyosin, and troponin complex, of which you can see here. You have the actin that has got the binding site, so the active site that is covered by the tropomyosin, a filamentous thin filament. Then we also have the troponin complex. Troponin complex is connected to tropomyosin. Just to expand on those for you to appreciate even more, so you have the troponin complex that is attached at a different segment on tropomyosin. The actin, it's a filamentous actin because there is polymerization of globular actin into the filamentous actin. So the interaction is inhibited by the tropomyosin that are covering the binding sites. So the troponin complex is composed of three protein subunits. You have the troponin T, I, and C, or troponin TIC, TIC. So T is a protein subunit that will anchor the troponin complex to tropomyosin. So troponin T is attaching the troponin complex to tropomyosin. The troponin I is for inhibition. So it's going to inhibit the activities between the actin and myosin. Then the troponin C is the binding site for calcium. So it's a receptor for calcium. So calcium can come and bind to troponin C, causing the troponin complex to change shape, pushing away the tropomyosin from the binding sites. Once the binding sites are exposed, then there will be interaction between actin and myosin that will bring about skeletal muscle contraction. But remember, we are just looking at the structure. The mechanism you appreciate in lecture 2, muscle physiology. So we continue with the structure. So now let's look at the sarcotubular system. So in skeletal muscles, the sarcotubular system are called triads because there are three structures that are contributing to form the sarcotubular system. In the cardiac muscle cells, the sarcotubular system is called the dyad because there are two major structures that are contributing to the formation of the sarcotubular system. But this is skeletal muscle. So let's appreciate the triad, which is forming the sarcotubular system in skeletal muscle cells. So we have two structures there, the sarcoplasmic reticulum and the transverse tubule system. So the muscle fibrils are surrounded by vesicles and tubules. So these structures or membranes form the sarcotubular system, which is made up of a T system and a sarcoplasmic reticulum. So they are made up of a T system. T stands for transverse tubule system, and the sarcoplasmic reticulum. The terminocystini or the sarcoplasmic reticulum, they are forming membranes that will contribute to the sarcotubular system. The T system or the transverse tubules, which is continuous with the sarcolemma of the muscle fiber, forms a grid perforated by the individual muscle fibrils. So the T system is more like continuous with the sarcolemma. So they are more like invaginations into the myofibrils. That will help to spread the action potential into the myofibrils. So the space between the two layers of the T system is an extension of extracellular space. So it's more like an extension of extracellular fluid compartment into the myofibrils because they're more like tubes that are penetrating the myofibrils, responsible for transmission of an action potential, the inward spread of an action potential. So the word triad is due to the three structures that are forming the sarcotubular system. Sarcotubular system is composed of transverse tubule, shown in yellow here, and the terminocystine, shown in blue. So the terminocystine or the sarcoplasmic reticulum will have receptors. Then within the T system also have receptors. So how many structures are here? 1, 2, 3. So it's called a triad in skeletal muscle cells. So the T-tubule, they are more like tiny tubes that are penetrating the muscle fibers. So you can see on the sarcolemma here, you have these holes. So these holes are more like pipes that are invading the muscle fiber. So they are going to spread an action potential, because an action potential is being propagated on the sarcolemma, then it can spread into the myofibrils via the sarcotubular system, especially the transverse tubule system. Okay. So it's the same structure here, just appreciating the sarcotubular system. So you have the terminal cisternae or the sarcoplasmic reticulum. At the center, you have the transverse tubule system that will spread the action potentials. The composition of the fluid that is found within the transverse tubule system is identical to that of interstitial, which is part of the extracellular fluid compartment. So electron concentrations, when you compare intracellular versus extracellular, it's the same here. So the electrolyte concentration of sodium, chloride, calcium is just the same when you compare it to extracellular fluid compartment, because this is the extension of extracellular fluid compartment into the myofibrils. They are more like tubes. Okay, so going inside the transverse tubule, there are receptors. On the sarcoplasmic reticulum, there are also receptors. So what receptors are there? So there are two major receptors that I would want you to appreciate. There's what we call dihydropyridine receptors and ryanodine receptors. So the dihydropyridine receptors, the DHPR, these are dihydropyridine receptors. They are found in the transverse tubule system. Then the ryanodine receptors, the RYR, they are found on the sarcoplasmic reticulum. So they are also involved in excitation, contraction, coupling of skeletal muscle cells. So this is what is happening. Remember, there is an axon terminal that is ending on the somatic motor, that is ending on the motor end plate. So this is a muscle cell. So this is a presynaptic membrane. This is a postsynaptic membrane in skeletal muscle cells. They're called motor end plate. They will contain receptors for acetylcholine. Then on the sarcolemma, which is the plasma membrane of the muscle cell, there is extension via the T system. So this is the T system, T tubular system. Within the T tubular system, you have receptors, which are called dihydropyridine receptors. So they are called dihydropyridine receptors. They are sensitive to change in voltage. So they are voltage sensitive proteins. So in skeletal muscle cells, on the sarcoplasmic reticulum, we have the ryanodine receptors, which are also channels, can allow movement of calcium. So the sarcoplasmic reticulum is a cell organelle that can store a lot of calcium. The concentration of calcium in the sarcoplasm, it's almost zero. Remember, 0.0001. 0.0001 milliequivalent per liter. So in skeletal muscle cells, the dihydropyridine receptor, they are mechanically connected to the ryanodine receptor. If there is a wave of an action potential that is moving down the T-tubule, it can stimulate change in dihydropyridine receptors that will mechanically open the ryanodine receptors, and then calcium will be mobilized from the sarcoplasmic reticulum. It will move into the sarcoplasm. That calcium can go and bind to troponin C, causing change in the troponin complex, moving away the tropomyosin, exposing the binding sites. Then there will be interaction between myosin and actin. That will bring about shortening of the sarcomere, hence skeletal muscle contraction. But we're not getting there yet. Now I just want you to appreciate the two receptors that are found in the T-tubule and ryanodine, and the ryanodine receptors on the sarcoplasmic reticulum. So dihydropyridine receptors and the ryanodine receptors that are found in the sarcotubular system. So the story, you need to start from the motor neuron that will fire an action potential down the axon, stimulating the opening of calcium-gated ion channels that will bring about influx of calcium into the axon terminals or the somatic motor neuron. that calcium will go and bind to V snare, T snare, facilitating exocytosis of acetylcholine. Acetylcholine will go and bind to acetylcholine receptors. That will cause movement of sodium and calcium, sodium and potassium. So potassium will be leaving the cell, sodium will be entering the cell. But the movement of sodium is more as compared to the movement of potassium. So the sodium that is entering the cell will depolarize the sarcolemma to the threshold. Then sodium voltage-gated ion channels are going to open at the sarcolemma. Influx of sodium here, it will trickle down to the next segment, stimulating it to the threshold, opening sodium voltage-gated ion channels on the next segment. So once they open, sodium will enter, it will trickle down to the next segment. So that is how the action potential will be propagated via the T-tubule system. This protein will sense the change in voltage that is brought about by the action potential. Then it will cause opening of the ryanodine receptors that will open. Then calcium will leave the sarcoplasmic reticulum to go into the sarcoplasm. It will diffuse to the troponin complex. It will go and attach to the troponin C. The troponin complex will change shape, pushing the tropomyosin, exposing the binding site on actin. And then myosin can attach. Hydrolysis of ATP will cause the myosin hairs to pivot, pushing the troponin, pushing the thin filament towards the M line, shortening of the sarcomere. Then you have muscle contraction. Okay, so it's basically the same here, just showing you the dihydropyridine receptor that are mechanically connected to the ryanodine receptor in the sarcoplasmic reticulum. So this calcium is the one that will bring about muscle contraction. But you need an action potential. So neuromuscular junction, transmission at the neuromuscular junction, it will bring about excitatory postsynaptic potentials, which are called motor input potentials, that will be added at the sarcolemma, then depolarizing the sarcolemma to the threshold, it will fire an action potential. That will be propagated into the T-tubule, then stimulating the dihydropyridine receptors, which are sensitive to change in voltage. Then they will mechanically open the sarcoplasmic reticulum by opening the ryanodine receptors. Then calcium will move into the sarcoplasm. Then it will go and bind to troponin C, tropomyosin moving away, exposing the binding sites. Then there will be interaction between actin and myosin, muscle contraction. The other point that I want you to note is the difference between the dihydropyridine receptors and the ryanodine receptors in cardiac muscle and skeletal muscles. So in skeletal muscles, that's the first diagram on our left. So they have voltage sensitive ryanodine receptors. So these voltage sensitive is because they are being opened by the dihydropyridine receptors. So the dihydropyridine receptors, they are sensitive to change in voltage. The change in voltage is brought about by the action potential. So they are going to open now the ryanodine receptors that will cause calcium to move from the sarcoplasmic reticulum into the sarcoplasm, bringing about skeletal muscle contraction. But this arrangement is different in cardiac muscle cells, which is very important for you to note. So in cardiac muscle cells, we have the dihydropyridine receptors, but they are also a channel. So they are more like voltage-gated ion channels for calcium. So they are called L-type slow calcium channels. L-type slow calcium channels, those are channels for calcium. So once calcium comes and bind, once there's change in voltage, so that change in voltage, it will bring about this channel to change shape, and then it will open up to allow movement. So in cardiac muscle fibers, in the sarcotubular system, we are saying that there's a difference in the arrangement of dihydropyridine receptor and ryanodine receptors. So here, the dihydropyridine receptors, they are also channels. So when there's change in voltage in the T-tubule, the dihydropyridine receptors, which are also channels, they're going to open up. So they are more like voltage-gated calcium channels that will open up due to change in voltage. Then calcium will move from the T-tubule. Remember, the T-tubule contains a fluid that is identical to that of extracellular fluid compartment. So calcium there is high as compared into the sarcoplasm. So calcium will start moving from the T-tubule into the sarcoplasm. So this calcium that is moving into the sarcoplasm, it will go and bind to the ryanodine receptors. Once the ryanodine receptors are stimulated by calcium, they are going to open up and allow movement of more calcium from the sarcoplasmic reticulum into the sarcoplasm. This calcium will go and bind to troponin C, causing the tropomyosin to move away, exposing the binding sites. Then there will be myosin-actin interaction that will bring about the myocardial contraction. So the ryanodine receptors in the myocardium, they are referred to as calcium-induced calcium-releasing channel. So calcium-induced calcium-releasing channel. So it means that they are induced by calcium. Then once they are activated, they are going to open to allow movement of calcium. So once they are going to open, they allow the release of calcium. So calcium-induced calcium-releasing channels are ryanodine receptors that are found in myocardium in the heart. Even in the smooth muscle cells, they could be there, but they're not a lot. So we don't discuss them under smooth muscle cells. But here we discuss them. So why am I mentioning this? It's because there are certain drugs that can inhibit the L-type calcium channels. So they will have an effect on the myocardium contraction. They will minimize or they will inhibit the contraction in the myocardium. So if you have a patient with tachycardia, an increase in the heart rate, tachycardia, you can use calcium blockers. They will go and block the L-type calcium channels. That will prevent influx of calcium. So if calcium is not entering the myocardium, it's not going to stimulate the opening of the ryanodine receptors. There will be no calcium to cause contraction of the myocardium. So the myocardium is not going to contract. Then the heart rate is going to decrease. But the calcium blockers, they won't have an effect on skeletal muscle cells. For skeletal muscle cells, they don't depend on extracellular calcium for their contraction. The calcium is just mobilized from the sarcoplasmic reticulum. That doesn't depend on the extracellular calcium. So you need to know that because there are certain drugs that will work on the myocardium that it won't work on skeletal muscle cells because of the differences of the arrangement. Okay. So it's basically the same information here, the difference between skeletal muscle and the myocardium. So you can see in skeletal muscle, there is a mechanical connection between the dihydropyridine receptor and the ryanodine receptor. So once there is an action potential, it will stimulate the change in the dihydropyridine receptors that will cause the opening of the ryanodine receptors. In myocardium, it's different. There's no mechanical connection here. So for the ryanodine receptor to open, they will depend on the dihydropyridine receptors, which are channels for calcium. Change in voltage, they will open and allow influx of calcium. That calcium will go and bind to the ryanodine receptors, opening them to mobilize calcium from the sarcoplasmic reticulum. So these are called calcium-induced calcium-releasing channel in the myocardium. Okay, so I think for the first lecture, this should be enough. So this should be enough for the first lecture. So we are done with the first lecture in muscle physiology. Then next time I'll record another video. It will just be on the mechanism of contraction for skeletal muscles, because now you know the structure, you know what is involved. So now we need to go into the actual mechanism of skeletal muscle contraction. Then after that, we'll discuss the cardiac muscle because it's similar to that of skeletal muscles. Then towards the end, we'll discuss the mechanism in smooth muscle cells. Then we'll look at other properties of muscle physiology, like the energy sources for muscle contraction and relaxation, different types of muscle activities. You know, we have isotonic contraction, and then we also have isometric contraction of skeletal muscle cells. So what would be the difference? So after the mechanism of contraction, that's when we can discuss all that. Otherwise, thank you very much for your time. I hope you find this video interesting for your studies. Thank you very much. Until next time.